from the James Hunt Institute, who's been studying with Vivian Block, and just finishing up her PhD there.
Hi, good morning everyone. So my talk today is about one chapter of my PhD, which is focuses on the role of gene enrichment in mapping a nematode resistance gene in Tetraploid potato. Okay, so unlike America, so all my work is done in Britain, mostly in Scotland. So unlike America, we have PCN everywhere. And we also have all three pathotypes, so we have PA1, PA2, and PA3, unlike in America where you just have PA2, 3. You also, like has been said earlier, there is no one resistance gene that can control pallida, so the best way to create broad spectrum resistance gene is to stack multiple resistance genes into a single cultivar in hopes that whatever pathotype you have in your field, you'll get at least some level of resistance. So my work focuses on the PA1 pathotype, which isn't the main pathotype in British fields, but it still poses a risk, and work still needs to be done on it, because if you're a farmer that has it, you kind of care about having some resistance towards it. Luckily, there is one dominant, dominant resistance gene, which is active against PA1, called H2, and it's from the wild potato species Salina multidissectum, and it gives a really strong resistance phenotype to PA1 and also a lesser resistance phenotype to PA2-3. So to do my research, I needed some material. First of all, let's talk about tetraploids. So multidissectum is a diploid, and diploid genetics is really easy. But for my PhD, I did all of this in tetraploids, which is a lot more difficult. So as an example, if you do a cross between two heterozygotic diploid, you get a 1:2 to 1 ratio, so you get 3 resistant plants to every 1 susceptible plant. Whereas we have 4 copies of every gene in a tetraploid, so if you did the same heterozygotic cross between 2 tetraploids, you get a really nasty ratio of 1:8 to 18 to 8 to 1, so a 35 to 1 resistance to susceptible ratio, which is just horrible to work with when you have to do genetic analysis. So to make our lives a little bit simpler, we used a homozygotic susceptible with a heterozygotic resistant parent, and you get a nice 1:1 susceptible to resistant ratio, which makes at least your phenotyping a lot simpler. So for this, we then had to find a resistant and susceptible parent plant, and for the susceptible donor, we used Picasso, which is a susceptible plant that we have in Britain, and for at least the last five breeding generations, there has been no sign of any sort of PCN resistance that we know of. And for the resistant parent, we used a breeding line called P557, and one of its grandparents was actually the wild diploid multidissectum species. So we know for a fact that this breeding cultivar definitely has the resistance gene within it. So using these 2 cultivars, we did a breeding to create a segregating F1 population. These population then went under 2 infection assays with PA1 cysts. So you just put your plant in a pot, put cysts in the pot, leave it for about 7 weeks, and then open up the pot and count the number of cysts that you see on the roots. We then plotted the infection and took the 20 most resistant and 20 most susceptible progeny plants for further analysis. So this further analysis was done using gene enrichment. So for the H2 gene, we had no genetic information about the resistance at all. From the screen, we knew that it was a simplex resistance gene, but we had no idea where in the genome it was. So to try and make our life a little bit simpler, right from the start, we used gene enrichment, which decomplexes the genome and lets you like narrow into the regions that you would want to look at. So we did 2 types of enrichment. So the first one was GenSeq, which enriches low copy genes, which are across all 12 chromosomes. And the second one is called RenSeq, which just enriches all known R genes. So we know there are 755 R genes in the potato genome, and this technique allows you to enrich for them all. So for the enrichment, you take your DNA and you fragment it like you normally would to make DNA libraries. But then you do an extra step where you mix it with bioaccumulated probe. So these are the probes that target the genes that you're looking for. So either the low copy genes or the R genes. And then because they're bioaccumulated, you can pull them out using streptobadin beads. So you only have the fragments of DNA which you hope will give you some useful information. And then, like any normal sequencing library, you do amplification and then resequenced using MySeq. So we created libraries for the susceptible and resistant progeny bulks, as well as the resistant and susceptible parents. So for the sequencing, when we got our sequence back and we assembled it, what we were looking for was differences, something that was in the resistant that wasn't in the susceptible, which could potentially be the reason that we see a resistance phenotype. So for this, we did SNP filtering. And basically, we get an output like this. So the top graph is what we got from the RenSeq, and the bottom one is what we got from the GenSeq. So for here, you've got all 12 chromosomes, and all the spikes show the density of where the probes are for the genes that we enriched for. And as you can see, there's no bias for a certain chromosome. They're kind of uniform across all 12. And each dot represents a SNP, and depending on how far up the axis depends on how many SNPs it's worth. So one down at the bottom is worth 1 SNP, where one up the top is worth 20, 30, whatever. And based on the filtering parameters that we used, it became very clear that the H2 gene is probably most likely on chromosome 5, and in particular in this 9 megabase pair region at the top distal end of the chromosome, which is great. And both enrichments kind of backed up the other, so we know it's probably chromosome 5. So the next step was to create some markers. So for each of the SNPs within this region, we created a set of KASP markers. So these are competitive allele-specific primers, which are made up of 3 primers. So you have 2 competitive forward markers and 1 universal reverse. And depending on the SNP which is present, one or the other forward primers will anneal. So once you do elongation, they're fluorescently tagged with a hex or a FAM. So you see a fluorescent output on the qPCR machine. And hopefully, if the allele that you have chosen, so the SNP that you chose to make your primer, can actually differentiate between a resistant and susceptible allele, you get nice clustering. So all your susceptible plants cluster in one, and then all your resistant cluster in the other, which is great. But sometimes the SNPs that you choose pass filtering for some other reason, and they're not actually to do with your phenotype. So you get this kind of mess where the computer just can't differentiate what is susceptible and what's resistant. But based on the SNPs that we filtered, we got a 75% success rate in marker design and development, which is great. So for the 75% of markers that we now have, we screened our segregating population to see what the genetic makeup looks like. So these are our markers. So the top line is at the top of the chromosome, working down. So it goes from about 1.4 to 11 megabase pairs. And anything that is green is resistant, anything that is pink is susceptible. And as you would expect, most of the resistant plants show the resistant allele, and most of the susceptible plants show the susceptible allele. However, there are some that show a mixture of resistant and susceptible, and these are recombinants, which allow us to map where our art gene probably is. So based on these plants, we could narrow it down to that the H2 gene is probably within this 4.1 base pair region, between about 2.2 and 5 megabases, which is great. But these 40 plants, this was the amount of it. We couldn't get any more information from these 40 plants, so we needed a larger population. So the next step was to use an expanded population of plants from the same F1 population. We used 650 plants and screened with these flanking 5 markers to find some recombinants. We did, out of the 650 plants, we found 65 recombinants, which we then phenotyped so we could link their genotype to their phenotype and then sort them and order them. So based on the 65 recombinants, we identified that the gene is now probably within 0.8 megabase per region, somewhere between 2.2 and 3 megabases, which is great. So the next step was to make sure that there's actually resistance genes within this area and that we've not just mapped something completely different to what we were looking for. And within the DM reference at least, there are actually 2 resistance genes, which made me very, very happy when I found these, which will then be taken forward as candidates for knockdown, knockout experiments to see if either of them are actually the H2 functional art gene. But what's actually the point in this? While this was all going on, we also started doing some breeding. So using the H2 resistant donor plant P55Z, we crossed that with a breeding line called 10Z3AA, which contains H1, which is a broad spectrum resistance gene against globodera stockensis, and H3, which is another pallida resistance gene. So the plan was that any progeny that were produced by this would hopefully have H1, H2, and H3, and would have resistance against all PCN. So the progeny were screened with the 57R marker, which is the marker that we have for H1. Progeny that were positive for that marker were then screened in a pot test using PA23 and PA1 to hopefully generate some material which has all 3 markers, which can then be taken forward into further steps in the breeding program, because what we really need is resistant, broad spectrum resistant cultivars. So I'd just like to say thank you. Thank you to Global mostly, because they're the ones that invited me and paid for me to come and speak here. So I'll take any questions.
Are there any questions for Shauna? Thank you so much, Shauna. Sorry.
Just wondering, with your 2 candidate R genes, do they have any homology known R genes besides like NBS and LR domains?
Yeah, so they're concoil NBS-LRRs, but they're completely different to the other 755. They're really close to each other, but nothing like any of the others, which is interesting.
Are there orthologs in Arabidopsis or any monoplants?
I haven't looked at that yet, but that's something